Hello student, I am Dr. Anupam, Department of Botany, Jamshedpur Cooperative College, Jamshedpur, Kolhan University. Today I am going to discuss about mechanical tissue. Mechanical tissue are special types of tissues that provide mechanical strength or support to the plants or plant parts. It means they are concerned with providing mechanical strength, rigidity and hardness to the plant body or a particular plant organ. Haverland in 1914 called the mechanical tissue as a stereome. Root or stem faces various kinds of strain of soil or air environment. Such strain causes them to develop strong mechanical tissue on various distributional pattern. That's why small land plants and water plants have poorly developed mechanical tissues, whereas longer land plants have well developed mechanical tissues. The chief mechanical elements are collenchyma, sclerenchyma and parenchyma. Today we will discuss about the structure of collenchyma. Collenchyma derived from Greek word colla or glue. It is restricted only to aerial parts. It is rarely found in roots and absent in monocots. They developed in roots when they are exposed to light. They consist of living cells, but pectic substances are deposited at the corners of cell present in outer region of cortex. Cortex of stem, leaves, petiole or of dicot plant. Collenchyma is closely related to parenchyma. It has thicker walls and longer cells. It differs from sclerenchyma in the presence of protoplast at maturity and in wall structure. Collenchyma cells vary in shape and size. The smaller cells are prismatic, while the elongated ones are usually with oblique or tapering ends. Individual collenchyma cell may often attain length of 2 mm. The wall of collenchyma are chiefly composed of cellulose and pectic compounds and contain much water. The thickening on the walls of collenchymatous cells are unevenly distributed. Normally, the wall thickening is present in corners. These are the wall thickening. They contain cellulose, pectin and hemicellulose. They are closely packed and may or may not have intercellular spaces. When intercellular spaces present, these are filled by pectin layers. Types of collenchyma. On the basis of thickening of the cell wall and arrangement of cells, Muller in 1819 gave 3 types of collenchymatous cells. These are angular collenchyma, lamellar collenchyma and lacunar collenchyma. But Duchesne in 1955 recognized one more type, that is annular collenchyma. The first one is angular collenchyma. It is most common type of collenchyma where the thickening are primarily at the corners or angles of the cell. In the figure of A, it is this transverse section of stem of Solanum tuberosum, and the figure B is the longitudinal section of stem of Solanum tuberosum. This type of collenchyma the cells of this type of collenchyma are compact and irregularly arranged. In this section, thickening are seen to be in those places where 3 or more cells meet. This type of collenchyma found in stem of Dhatura, leaf petiole of Vitis, Begonia, Cucurbita, stem of Solanum tuberosum, Atropa belladonna, Dahlia, etc. The next one is lamellar collenchyma. In this second type of collenchyma, thickening are mainly on tangential wall of the cell. In this type of collenchyma, cells are compactly arranged without intercellular spaces. This type of collenchyma tissue is found in stems of Sambucus nigra, Helianthus, etc. The third one is lacular collenchyma. In this type of collenchyma, the thickening appears in those parts of the cell wall that face intercellular spaces. The thickening appear at the intercellular space facing part of the cells. This type of collenchyma is also called tubular or lacunate collenchyma. This type of collenchyma found in petiole of Salvia, Cucurbita, Malva, and in the members of Compositae family. The fourth one is annular collenchyma. Here is the thickening of the cell wall is angular fashion, that is around the entire cell wall, so that even cell loses its identity, in which the cell's lumen is almost circular in cross section. This type of collenchyma found in Nerium oleander. That's all for today, students. In the next video, we will discuss the origin, distribution and function of the collenchyma. Thanks for patience listening.